This is lab number 3 that I'm working on right now. It'll be posted at the end of this week. I believe it's the 28th today, Friday, so I'll have it up next week. We'll be working on mark and recapture till towards the end of next week anyway, so maybe we'll get this one up early. We'll see. This is an indoor lab. You'll notice there's no puppy, Mr. Squiggy, in here. This one's one of the ones we normally do in the classroom. You'll look on your lab folder for lab number 3. It's a metabolism lab. You'll have a handout, the one we normally use in lab. So I'm about to go through the procedure. Really, there's not going to be that much for you guys to do. Once I've done this video and gone through the procedure, you're just going to have to make me like 3 figures, graphs. That's all you're turning in. So it's really not too bad. Again, this is a metabolism lab. If you've had 112, you've talked about metabolism a little bit. If you're in 112, you may get to it later in the semester. But metabolism is basically how organisms pull the energy out of their food items to use that energy to fuel their metabolic process. Active transport pumps, motility of some sort, flagellus, cilia, muscle contraction, all of that. Now, you'll go over things like glycolysis and the Krebs cycle and electron transport chain, stuff like that. All we're looking at today in this lab covers one small aspect of an energy liberating process. It's called fermentation. Fermentation is a metabolic process that allows organisms to get energy out of their organic molecules, their food, in the absence of oxygen. It's fairly inefficient, but under anaerobic conditions, that is when there's no oxygen available, it's about all you've got to use. So fermentation in humans would go along with what they call glycolysis. It works very similar in humans as it does in many other animals, but today we're using yeast as an example. They're a fungus, not an animal, but they also have a fermentation process. It's slightly different than ours. We use what's called lactic acid fermentation. The yeast use ethanol fermentation. So it's an important metabolic process biologically. It's also got some importance economically for people. Yeast fermentation is used for any of your alcoholic beverages. This is how they're produced. Beer, wine, your distilled beverages, whiskey, vodka, what have you. They're all made through ethanol, yeast ethanol fermentation. Also, the E85 fuel you might buy at the speedways in town. That's all the ethanol for fuel. It can be used as a motor fuel. Is also made using this process, as well as all your hand sanitizers. Many of those that they're using right now are like 60–70% ethanol. It's made from ethanol fermentation. First, let me give you kind of a quick equation. What these organisms will do is they take a food source. This C6H12O6, it's glucose. It's a carbohydrate. That has energy in it, potential energy in the chemical bonds or covalent bonds holding all these atoms together. Now, in fermentation, you don't need any oxygen. The fermentation pathways allow you to break this glucose down and produce, as sort of a byproduct or end product, carbon dioxide. Again, these are all the atomic symbols. You're working on them in 112, or you already have carbon, oxygen, hydrogen. This stuff right here is going to be important for our lab. Let me get a marker that actually works. CO2 is carbon dioxide. It's an end product of fermentation. It's also an end product of cell respiration. We exhale it. In high concentrations, it is toxic. But this carbon dioxide is a waste material. It's produced as a gas. It's going to be released in the atmosphere. And this stuff right here is what we want to keep. This is ethanol. Now in fermentation, the ethanol is also a waste product. If you put these yeasts in a container, they'll break the sugar down, producing carbon dioxide and ethanol. Carbon dioxide will diffuse out into the atmosphere. The ethanol builds up in the solution. Eventually, ethanol is toxic. That's why we use it in hand sanitizers, for example. At about 12% or more, it starts to kill the yeast. So there's basically these yeast are eating all the sugar, and they're more or less swimming around their own waste material, this ethanol. When it builds up to a certain point, it'll kill them. That's why things like beer and wine typically aren't much worried about 12% ethanol. You put yeast in a container with some kind of carbohydrate, sugar, whatever source, and you can only get to about 12% or so ethanol, then the yeast die, that's all you got. Now today, we're going to be taking these yeasts, we're going to be feeding them a carbohydrate source. It's sucrose, not glucose, but fundamentally it'll work the same way. And we're going to monitor a few variables that might affect how fast fermentation occurs. So during this experiment, we want to see how much fermentation is occurring. We're not going to measure the ethanol, although you can. It's just technically more difficult. We're going to actually use CO2 production to get an indication of how much fermentation occurred and how much energy was converted by these yeast into a usable form. The way we're going to do that is we're going to set up, it's kind of an ugly flask, but this represents a flask. It's just a glass container. And we're going to put in this container some water with sugar, will be sucrose, just table sugar, and yeast. And then what we're going to do is we're going to stick a balloon on the top of this thing. Now, one of the assumptions we're making, as fermentation occurs in this fluid, the CO2 that's produced is going to be released as a gas and inflate the balloon. Now, it's kind of a crude measure, so it's just kind of maybe useful for relative comparisons. The bigger this balloon gets over time, we can make the assumption that more fermentation has occurred. So the way we're going to measure fermentation is by measuring basically the circumference of this balloon. The bigger it gets, the more CO2 is produced, the more fermentation occurred. All right. Now for the experimental setup. What we're going to do, we're setting up 6 different flasks. In your data sheet, I'm going to refer to it as I set them up. They're called group 1, 2, 3, 4, 5, and 6. So for group 1, what I'm going to do, I'm going to set up 1 flask. It's an ugly flask. And in that flask, I'm going to put 3.5 grams of yeast, no or 0 grams sugar or sucrose. So that flask will have some warm water in it, yeast, no sugar. Then I'll go ahead, mix it up, put a balloon on it, and I'm going to set it on the table out here at room temp. Flask number 2, or what we call group 2. Set up the same thing. I'm going to take a flask. I'm going to put in 3.5 grams, I'm going to put a G there, of yeast. But this time I'm going to add some sucrose, some sugar. It'll be 10 grams of sugar. So that's the second flask. Set it up, mix it up, put a balloon on it. Group number 3, also 3.5 grams of yeast. In fact, every flask is going to have 3.5 grams of yeast. That's not going to vary. But the sugar is going to be 40 grams. So notice all I've changed with flask number 1, 2, and 3, they have the same amount of yeast. They're going to all be at the same temperature. This is considered the room temperature group. It's 20 degrees Celsius in this room. One flask, number 1, has no sugar. Flask number 2 has 10 grams of sugar. Flask number 3 has 40. Then I'm going to set up 3 more flasks. Flask number 4, it'll have 3.5 grams of yeast. It's going to be true for all of them. And it'll have 0 grams of sugar. But this one is going to be put in a warm temperature bath. We'll keep it at 37 degrees Celsius. I'm sorry, it's not 37. They got it set at 35. So we'll have 3 flasks at room temperature, 1 with 0, 1 with 10, 1 with 40 grams of sucrose. We'll have another 3 flasks, number 4, number 5, and number 6. They'll all have the same amount of yeast, all be at 35 Celsius. So it's pretty warm, it's close to body temperature. But again, grams of sugar will vary. So here's what you have to look out for. All right, we're going to manipulate some variables. We'll call them independent variables to see what effect they have on fermentation or CO2 production. which is the sort of independent or the dependent variable. So we are changing temperature. You've got two different temperatures. Some of these flasks will be at room temperature, about 20 Celsius. Another three flasks, 4, 5, and 6, are going to be warmer. They're at 35 degrees. That's the difference between these two groups. The only other thing we're varying is the amount of sucrose or sugar in the solution. Some of the flasks have 0, some have 10, some have 40. So you've got two variables we're manipulating. We're really sort of two experimental treatments, I guess is one way to look at it. We're going to change the temperature, or we're going to change the amount of sugar. And we're going to see what effect changing temperature or changing the sugar concentration will have on rates of fermentation. That's all we're going to set up. Now when we're done, we're going to take our one sort of dependent variable, how much, basically it's the circumference of the balloon, how much CO2 is produced. I'll put that on a table. Let me show you the table. It's in your lab book. I'll put it up here on the board and see what you have up here. You've got that's flask 1, 2, 3, 4, 5, 6. So with these three here are at room temp. These three here are at 35. And then that's 0 grams of sugar, 10 and 40, 0, 10, 40. So you can get what's in these flasks right off the data sheet. What I'll be filling in here, this is time. So initial time is just when we start the experiment. And then every 15 minutes, so at 15 minutes, 30, 45, 60, 75, I'm going to measure the circumference of the balloon. So the numbers that will be in these squares will be the circumference of the balloon over time. That's what we're monitoring to figure out how much fermentation occurred. So I'm going to set up the experiment now. And I'll just show you real quick what we've got set up here. This is what will go into the flasks. So this is flask number 1. It's labeled. It'll have 3.5 grams of yeast and no sugar, no sucrose. Flask number 2. Again, all the same amount of yeast, 3.5 grams. This one will have 10 grams of sucrose, sugar. Flask 3 will be set up. Again, 3.5 grams of yeast, 40 grams of sucrose. Flask number 4, the yeast, no sucrose. 5, again, the same amount of yeast, 10 grams of sucrose. And number 6, so 3.5 grams of yeast, 40 grams of sucrose. And remember, 6, 5, and 4, I'm going to place in an incubator I've got set up over here. This is a warm water bath. It's temperature controlled. About 35, it's reading 35.6 degrees Celsius. So that's where 5, 6, and 7, or 4, 5, and 6 will be. 1, 2, and 3. That one, and that one. They're just going to be out on the table. We'll let them ferment at room temperature. So I'll put a few more short clips on here of the balloons expanding due to carbon dioxide production. Hold on. All right. These are the setups. There's your flask number 1 with the balloon. That's 2 and 3. They're going to stay on the table. 4, 5, and 6. I am now going to move to the incubator. But before I do that, we need the initial measurement. So I'm going to take this tape and for the initial measurement, all I'm going to do, see if we can leave it on the table there, so a better view here. All right, I'm going to take this tape. I'll use centimeters here, the millimeter or metric side of this thing. And I'm going to just go around kind of the fat part of the balloon. Of course, it's empty right now, so the circumference is going to be tiny because there's nothing in it. So it looks like it's 2 centimeters. If I squeeze this together, I'm just going to call it about 3 centimeters. So the circumference of the balloon was right about there. That's my initial measurement. I'm going to do that for each of the flasks And then we'll wait every 15 minutes and record it again. All right, this is after 15 minutes. Flask number 1 didn't change really anything significant. Flask number 2, there's a little gas in there. It grew a little bit. Flask number 3, not much change yet. Let us go to 4, 5, and 6. Let's see, 4. Yeah, a little bit. Of course, this might have expanded a bit too. I don't expect much to happen in 4, but a little. Ooh, 5. 5 is cranking up there. It's starting to inflate the balloon. And 6 has a bit of gas in it as well. Now, the data, this is what you'll end up using to do your reports or your graphs. So when this is all done, I'll go ahead and type it up and post it on the Blackboard site. But for example, you know, there's balloon or flask number 5, started at 30 millimeters. It's now 160 millimeters. All these units will be millimeters. You know, flask number 1, 30 millimeters. After 15 minutes, still 30. Flask 2, 30. It went to 60. So you'll have that data posted on Blackboard. That's it for now. All right. It's been 30 minutes now. That's number 1. No significant change. Number 2 is inflating a little bit. Number 3, and didn't notice much difference there. Those are the room temperatures, flask 1, 2, and 3. Number 4, 5, and 6 are in the incubator 4. Yeah, not much of any change there. You know, a little bit of gas in there. Could be from expansion. That's number 5. It's looking pretty big. It's grown quite a lot. And that's number 6. It's getting some gas in there as well. Take a break for another 15. All righty. That is your data. Now, I know you can't read it well on here. That's all right. This will be typed over this table. I'll fill it out. That'll be posted on Blackboard as your fermentation data. It'll just be a word page. Now, what do you have to do? Let me give myself some clear whiteboard. We'll go over here. All righty. You read through the lab sheet, got the data, this is it. It's simply going to ask you to make 3 figures, 3 graphs. That's all you have to do. You're going to graph this data 3 different ways, upload it, you're done. It's the quickest 5 points so far. Not that much to look at. The first graph you'll have to do is, I want you to take all this data in some form, and keep in mind you have some flexibility here. You know, the type of graph you use a little bit can change. You know, how you arrange it can be different. It's all right. Remember, with graphs or figures, the idea is to visually show the data so someone can look at it quickly and go, oh, here's what's happening. You know, try not to make it too confusing or 2.5D. Simple is often better. But for this one, I believe it asks you to do a line graph with all the data. The x-axis and the y-axis. Typically, the y-axis is going to be, that's a dependent variable for pretty much everything. How much CO2 was produced? Our indicator for how much CO2 was the circumference of the balloon. That's what your data points are. That's the circumference of all the balloons in millimeters. And so you have 6 different flasks. So if you're plotting all the data, you need them all in there. Now, again, there's several ways you can do it. You can color code it. For example, let's see, flask number 1. The x-axis will be time. That's kind of standard for a normal graph. We got 0 here, initial. We got 15 minutes, 30, 45, 60, 75, something like that. So those will be your points. I guess I shouldn't put the line in first off. But if you look over at your data, this is circumference. You can start at 0. Our maximum value is about 3 something, so I'll go 320. So let's say this is 30. Balloon number 1 was started at 30 millimeters circumference, and at 15, 30, it didn't change. So you might have a line like this, basically a horizontal line. And you need to code it. So green equals flask or group number one. Then you switch colors or pattern. You use dotted lines, dashed lines, thick lines, skinny lines, different colored lines. You have some other balloon. And it may start about 30, but I don't know what they did. You know, go something like this. And then you might have another one. Go like this. And you might have a third one. Go like this. You'll have 6 lines. This is a time, and you'll have a key down here. You know, group 1, group 2, group 3, whatever they are. Label them. That's it. That's one of the 3 figures. The other 2, you have some options. It tells you to pick basically any of the independent variables, which for us I'd use, you don't have to, but you've got glucose or sucrose concentration, the sugar concentration, and you have temperature. Make a couple bar charts. I think in the instructions it says make a bar chart. You have to average things together. For example, you had 3 balloons that we incubated at room temperature, 20 degrees, and you had 3 incubated at 35. So those are your 2 points here and here. Again, y-axis CO2. Take all 3 balloons at room temp, take the average circumference for the balloons, probably the final circumference, but you know, you could pick dots in between. Again, there's several things you could do. Then you take the 3 class or groups that were in the incubator, get the average for their CO2, or circumference, I should say. There you go, bar chart. That would be chart number 2. Chart number 3. Do something similar, except don't use temperature. You could use the amount of sucrose you added. In that case, we have 3 different amounts of sucrose, 0, 10 grams, or 40 grams. You have 2 at 0 grams, 2 at 10 grams, 2 at 40 grams. Average those 2 together, and see what your charts look like. I don't know what they'll look like. Who knows? That is all you have to do. I will ask you to plot these again. Use the Excel plotting program or the little drop-down insert data table in Word. That's easiest. That way you can upload them as an Excel or Word file, and that's easier for me to open up and take a look at. Great. That's all you have to do with this one. So you got the data, it'll be up in the data table. It'll say fermentation data. Draw me 3 figures, 5 points. Take care.